We're Group 8 and we present you with chloramphenicol.
Chloramphenicol is a broad-spectrum antibiotic and is used to treat bacterial infections. These can include superficial eye infections, bacterial infections in the otitis externa, life-threatening infections caused by Haemophilus influenzae, and thyroid fever. In practice, chloramphenicol is mostly used to treat superficial eye infections. Chloramphenicol is available in many forms, including eye ointment, eye drops, ear drops, capsules, solution for injection, and IV infusion. These are typically only used in life-threatening situations. Chloramphenicol was first discovered in 1947. It's produced by cultures of Streptomyces venezuela from soil. In 1948, American medical researchers Theodore Woodward and colleagues published the first report on the use of chloramphenicol for treatment of typhoid fever. It revolutionized the field by reducing both morbidity and mortality. This was also the year chloramphenicol was incorporated into clinical practice under the trade name chloramycetin. Chloramphenicol became the first antibiotic to be manufactured through large-scale chemical synthesis. Later on, chloramphenicol was linked to rare cases of aplastic anemia and fatal blood dyscrasias. In the early 1960s, more than 1,000 cases of severe bone marrow aplasia were attributed to the use of chloramphenicol. As a result, chloramphenicol was either banned or placed under restrictions in usage ever since. In 1952, Ddmda, a structural analogue of chloramphenicol, was first synthesized. The nitro group is replaced with a methynyl sulfonyl group in Ddmda. While it doesn't cause fatal aplastic anemia, it can cause reversible toxicity in bone marrow and kidneys. However, it was found that the antibacterial activity was weaker in Ddmda than it was in chloramphenicol. Due to this, Ddmda is now primarily used for veterinary practice. Sparsifenicol, discovered in the 1980s, was the first chloramphenicol hybrid that bonded with a thiamine. In vitro studies suggested that sparsifenicol, at minimum inhibitory concentrations, strongly inhibits the puromycin reaction, which is important for transpeptidation. However, sparsifenicol is not lipophilic enough to penetrate the bacterial envelope, so it cannot inhibit the synthesis and the growth of bacterial cells, such as E. coli. Therefore, investigation of the drug was suspended. Florphenicol was discovered in the early 2000s. It's synthesized by replacing the position 3 hydroxyl group of chloramphenicol with a fluorine atom. Florphenicol was found to be a broad-spectrum antibiotic in vitro. Its antibacterial ability is as strong as chloramphenicol, but it also combats chloramphenicol-resistant strains and has much milder side effects. In vivo, florphenicol was reported to penetrate well into cerebral spinal fluid, which shows promising pharmacokinetic properties to treating meningitis. However, florphenicol cannot deal with strains bearing resistant genes of drug exporters and mutations. It's expected that florphenicol can be extended to clinical trials in the near future.
So, what does chloramphenicol actually look like?
Chloramphenicol has 2 alcohol groups, 1 benzene ring, 2 chloro groups, 1 nitro group, and 1 amide group. Overall, chloramphenicol has 5 hydrogen bond acceptors, 3 hydrogen bond donors, a molecular weight of 323.126 grams per mole, and a partition coefficient of 1.1. This means that it fits Lipinski's rule of 5. Chloramphenicol is perfectly fine to be administered orally. However, it can't be infused as it has a pKa of 7.8. By converting the alcohol group closest to the amide into an ester with a carboxylic group, chloramphenicol succinate is made. The pKa becomes 3.68 and its solubility is increased greatly. The functional groups we just covered contribute to chloramphenical's antibacterial activity.
How was chloramphenicol manufactured?
One of the first methods of synthesizing chloramphenicol involves condensing para-nitrobenzyl chloride with ethyl melanate, and the steps produce a racemic mixture. Manufacturers usually need to perform additional steps to separate the stereoisomers. During the 1960s and 70s, a new manufacturing method was developed in Western Europe. This didn't require additional steps to separate the stereoisomers. This method involved condensation of benzaldehyde using 2-nitroethyl in basic conditions, producing nitrodiol. The nitro group in nitrodiol is reduced to 2-amino, 1-phenyl, 1,3-propanediol, which is acetylated using acetic anhydride, for which pyridine is a catalyst. The acetylation product is nitrated using concentrated nitric and sulfuric acid. The product is then hydrolyzed to remove acetyl groups. Through swing resolution using tantric acid, the racemic mixture is separated and the stereoisomer is acetylated with dichloroacetyl chloride at the amine to produce a higher yield chloramphenicol. The activity takes place in 50S ribosomal subunits in bacteria. Chloramphenicol inhibits peptidyl transferase activity and hence inhibits amino acid containing tRNA binding to A site, therefore inhibits protein bond formation. Chloramphenical's interaction with the peptidyl transferase is attributed to different functional groups. First, the hydrogen bond donors and acceptors form hydrogen bonds with various nucleotides of the polypeptidyl transferase cavity. Second, the phenyl group forms pi-pi stacking interactions with the cytosine ring of the polypeptidyl transferase. In addition to these, putative cations, magnesium C1 and 2, a mediator, further stabilize the bonding between chloramphenicol and the polypeptidyl transferase cavity by facilitating the bonding of the 3' hydroxyl group and the para-nitro group at the peptidyl transferase cavity. While chloramphenicol does bind to the 50S subunit in eukaryotic cells, it's to a much less extent. This is why chloramphenicol won't kill you at the same time.
I see. Then can you tell me more about the side effects of chloramphenicol?
Several side effects come with systemic administration of chloramphenicol, one of which is hematological toxicity in the bone marrow. This can be reversible or irreversible. Reversible blood marrow suppression is dose-related and predictable. Red blood cells are suppressed inside the bone marrow. It's probably due to the inhibitory action of chloramphenicol on mitochondrial protein synthesis, when chloramphenicol plasma concentrations are 25 microgram per ml or higher. This is often observed in patients under the use of large doses of chloramphenicol, prolonged treatment, or both. In irreversible bone marrow suppression, chloramphenicol can cause aplastic anemia, fatal pancytopenia, leukopenia, and thrombocytopenia. The exact mechanisms of these are uncertain at the moment. It is, however, suspected that chloramphenicol-induced aplastic anemia is related to DNA damage caused by metabolites in bone marrow. Another severe side effect is grey baby syndrome. The manifestations in the first 24 hours are vomiting, refusal to suck, irregular and rapid respiration, abdominal distension, periods of cyanosis, and passage of loose green stool. Soon they become flaccid and hypothermic. And finally, the most characteristic symptom is that the baby develops a grey or blue skin colour due to poor blood circulation. This syndrome is caused by excessive or prolonged doses of chloramphenicol in newborn babies, especially if they're premature. The underlying reason why chloramphenicol can cause this is that chloramphenicol is metabolized through phase 2 metabolism by undergoing a conjugation reaction in adults. This reaction is aided by UDFP gluconorate, an enzyme which aids in the addition of a gluconoric acid molecule to the indicated hydroxyl group, resulting in chloramphenicol gluconoride. After this, the metabolite can be excreted in urine or bile. However, this reaction doesn't take place in babies, so the drug cannot be excreted out of the body. This leaves it to accumulate and eventually reach toxic levels in the liver.
I see. So to conclude, chloramphenicol is a broad-spectrum antibiotic that works by inhibiting peptidyltransferase in bacterial ribosomes. It is the first antibiotic to be fully chemically synthesized at an industrial scale. It is first-line treatment for superficial eye infections and bacterial ear infections. The possible side effects of chloramphenicol include the grey baby syndrome and bone marrow suppression.